Hi students! In this video we are going to look big picture. We are going to use this as an introduction to the cell cycle. So use this as a intro or an overview, and then you can go to the other videos that hone in on the different points of the cell cycle. Okay, let's get started. So first let's talk about the reproduction of an organism versus the reproduction of cells. Now, in the case of single-celled organism, the entire organism is the one cell. So if the one cell replicates, then you have essentially replicated the entire organism. This is called asexual reproduction or binary fission. Binary meaning two and fission meaning to split. So it splits itself in two. It has created an identical clone of itself. So you start with one, it duplicates everything on the inside, and then just kind of splits. Boom. Two identical clones. Now, in multicellular organisms, when that organism needs to reproduce, it's going to use a process called meiosis. This is called sexual reproduction, and because it uses sex cells, those sex cells of sperm and an egg. And each of those sperm and an egg have half of the amount of chromosomes that the organism has in a full cell. So the process of meiosis is creating those half or haploid, was what we call them, haploid cells. Those cells with half the amount of chromosomes, and that sperm and egg. And so to create that, we start with the parent cell that has, in this case, we're going to use humans for example, but that we start with the parent cell that has a full set of chromosomes. Humans have 46, and the process of meiosis creates two sperm cells here, two sperm cells that have half of that amount. So they have half the number of a total chromosome amount, so they have 23 each. So you have a 46 chromosome parent cell that is then meiosis creating two 23 chromosome sperm cells. And the same thing goes for an egg cells. And then that 23 chromosome sperm cell can bind and fertilize a 23 chromosome egg to where you have this 46 chromosome fertilized eggs that has a total full set of DNA chromosomes. This creates an organism, a brand new organism. And since the sperm and egg are different from that original parent cell, it had half the amount of chromosomes. And we're going to find out too in the process that you get a whole bunch of like mixing of those chromosomes, because when they make them, you get some cool uniqueness coming in. But that sperm and egg are different than the original parent cell. And so because they're different, then that offspring is going to be different. And so you get this unique fertilized egg. And so you get those, every one of those fertilized eggs is like 1 in a trillion combinations. And so that's what makes you amazingly unique and beautifully unique, because you are this amazing combination of unique chromosome matching. So it's so cool, and that's why everyone is beautiful in their own special way. Anyway, I digress. Now, if a cell needs to replicate itself completely, now we're not going to replicate the entire organism, we just want to replicate one cell. So say in the case of like you have a paper cut on your hand, and so you're like, oh no, like those skin cells are on, like, oh no, we have a paper cut, we lost some cells, we need to replace them, because you want to make sure you replace that those cut skin cells. And so that's where you're going to come in, the cell cycle, and with mitosis is going to come in, and it's going to create an exact duplicate cell. So this is where you have a 46 chromosome parent cell, and the process of mitosis and in the cell cycle is going to create two identical daughter cells that have 46 chromosomes. Again, this is humans, 46 chromosomes. Other organisms are going to have different total amounts, but we're just going to use this for simplicity. Now, so 46 chromosome parent cell, mitosis creates two 46 chromosome daughter cells. Now, I don't know why they call them daughter cells. I don't know why they call them son cells. I don't know, maybe that's because some cells is like a tongue twister. I don't know. But anyway, they're called daughter cells. So these daughter cells are completely identical. And again, like I said, as an example, if your skin gets a little paper cut, then you need to make an exact clone of itself. You don't want to have like some unique cell coming out of here. You don't want to have some liver cell being made here or a heart cell being made here. You want that skin cell. Whatever cell was lost, those other cells around it want to replace it with an exact duplicate of themselves. And so that's where this comes into play. Okay, so before we go on to talk about the cell cycle and to talk about like those steps of cell cycle, I just want to do a brief review of DNA and chromosomes. Now remember, I had a whole video just talking about DNA and chromosomes and how they're moving around in the cell. But if you recall, the DNA is typically in those long strands that are balled up like a yarn. So you've got these long stringy strands like yarn, and it's all in a big old ball here. And then when they are ready to divide and to replicate for mitosis, they then get condensed down into chromosomes so that you can have them replicating and then distributing really easily during mitosis. And so this is just kind of our sketch here of a single chromosome. You duplicate that chromosome, and then you get that really cool centromere that holds it together, like a little inner tube holding those little sister chromatids together. Again, sister chromatids, I don't know why they're not brother chromatids, whatever. We're going to go with it. But they're called sister chromatids. And again, that centromere here attaches to the center of them. Okay, so let's talk about the cell cycle here. It is going to happen in 3 stages. The first stage we call interphase. This is going to be the longest stage. So I would add that to your notes there. It's the longest stage. This is because during it, it's when the cell is undergoing its normal process. It's just chilling, being its own little skin cell, just hanging out there, one little skin cell here, just chilling. There, or you got a heart cell just chilling, being a little heart cell, or a blood cell just chilling and doing its thing, carrying blood oxygen, and picking up some CO2. It's just chilling and being a cool insulin-producing cell in the pancreas. I don't know, all the cells, just doing its thing. But when it's triggered that it needs to divide, then it's going to start growing a little bit, and it's going to replicate its DNA so that it can divide in mitosis. And then it's going to start dividing and making a replicate or duplicate of itself. So that replication and that dividing isn't going to happen just boom, boom, boom. Like here, we divide here, done. No, it's going to be 2 stages. 2 stages. You're going to first divide the nucleus, and then you're going to divide the cytoplasm. So mitosis happens first. So mitosis, you're going to divide that nucleus. You have the nucleus, you're going to divide it into 2 nucleus, and then you're going to divide that cytoplasm next. And you remember the cytoplasm, kinesis, moving. So cytokinesis. Okay, so why do we need to do the cell cycle? The purpose here, we already talked about one, is to replace those dead and injured cells. If you have a little paper cut, you want to replace that injured cell. Or sometimes cells are going to just wear out and they're going to die and they're going to need replacing. Like your blood cells, they only last, red blood cells only last about a month, and then they're going to need replacing. So they just constantly replicating and replacing those old, dead, injured, worn out cells. The other one is to grow an organism. And so when you're a lot bigger than when you were born. My kiddo was, now I can't remember, 8 pounds, 5 ounces when he was born, and now he's already 31 pounds. Ooh, my little big baby. And eventually he's going to be a tall little kiddo. Well, he wouldn't be a kiddo then he's tall eventually. You know what I mean? But you're growing. And so in order to grow, you're going to have to replicate those cells. You're going to have to make exact duplicates. Your bones are going to get longer. Your muscles are going to get longer. So you're making more of the exact same ones, but they're just getting so that the whole organism can get bigger. And so that's where you're going to have to grow and use the cell cycle. And you want every one of those bone cells in your femur to be exactly the same. So you're just going to keep replicating them until it gets to its final adult height. Now, the other thing that we're going to talk about is how. How does this happen? Number one is that this is very, very sequential. Every step has to happen in a specific order. So step 1, then step 2, then step 3. So mitosis has to happen before cytokinesis. And as you're going to delve into mitosis, and you're going to delve into those 4 substages of mitosis, each one of them have to happen before the next one. So it's going to have to go in very sequential order. I mean, and it only makes sense, really. So you can't split the chromosomes into 2 identical nuclei if you haven't replicated your DNA and replicated those chromosomes. You can't split them and make them identical if you haven't replicated them, right? Yes. So that just makes sense. So you have to go in a specific order. It's going to be very, very controlled. So when it's going in that order, it is not going to go to the next step until it is absolutely ready to go on to that next step, because there are going to be very important checkpoints. So before that cell can go from interphase into mitosis, or mitosis into cytokinesis, and then even within mitosis, before it can go into the next one, the cell is going to check to make sure everything is ready to go. Every part of that DNA has been replicated perfectly, and every part of that DNA has been arranged perfectly, and that centromeres are completely lined up, or that DNA has been completely divided evenly. I mean, everything has to be perfect before it progresses on. And the reason why they do this is that we want to make sure that that DNA is copied exactly. We do not want any errors. No errors in the DNA. You want to make sure that every cell gets the proper amount of DNA, and that DNA is exactly what it should be. Because the thing is that errors in the DNA are what we call mutations. And these kinds of mutations are not the cool mutations that give you superhuman powers. This is not going to give you Spider-Man powers, Superman powers, the ability to fly, or the ability for telekinesis or teleporting, which that would be like super awesome. But this type of mutation is not going to make you an X-Men. X-Men? X-Man. X-Men. You'd be part of the X-Men, so you would be an X-Man, but you could be an X-Woman. Ooh. I digress. I need to look that up. Anyway, this type of mutation is going to lead to bypassing those checkpoints. What that means is that your cell is going to go on to that next step. It is going to continue on that cell cycle and divide before it's ready to do so. That is not good, because that is going to lead to uncontrollable cell growth. When it starts replicating and dividing before it's not supposed to, it's uncontrolled. You're continually going through that cell cycle without any control. And what that leads to is cancer. And we are going to have a whole project on that, and we're going to delve right into that, like how the mutations lead to that and what that uncontrollable cell growth really looks like. And we're going to delve into that later. But just kind of for now, know that if we don't control this cell cycle, that's what we're going to get, and that's not good. So, all right, and I think that's it for this overview. There's already a video here about how those chromosomes move around, so then that's the end of our notes here. So, again, this was just an overview of the cell cycle, and now you can go delve into those stages of cell cycle. So thank you for watching, and always remember, be good, be kind, be awesome, and recycle. Bye.